Hello, everyone. Thank you for attending today's webinar, CROs, the tip of the spear in maximizing the value of hybrid, virtual, and synthetic clinical trials, presented by RO. I'm Danya, and I'll be moderating this webinar. Our speakers today are Rob Wilson, Chief Strategist of Biostatistics and Standards for Regulatory Submissions, and Derek Lawrence, Associate Director of Data Management. You can read their full bios on the left side of the window by selecting the Speakers tab. Just a few technical notes before we begin. If you'd like to download the slide deck, please click the Handouts tab on the left side of your screen. The webcast is being recorded, so there is no dial-in number. For the best audio quality, please make sure that your volume is up. The webinar will be available on demand within 24 hours after the event, and time permitting, we'll follow the presentation with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. Okay, now let's begin. Rob, please go ahead.
Thank you. Good afternoon. My name is Rob Wilson, and along with Derek Lawrence, who you'll hear from in just a moment, we'll be talking about and sharing our thoughts on maximizing the value of the decentralized clinical trial. As I'm sure most of you are aware, there has been a large volume of discussion around the clinical aspects of decentralized clinical trials, in particular, and rightfully so, focused on the benefits to patients of these trial designs. Derek and I are going to be talking about another aspect of decentralized clinical trials, and the focus of this talk will be on maximizing the value of the data that are collected from these ever-increasing number of data sources in these clinical trial designs. The challenge for us and for many others operating in this space is how do we handle the increase in volume of data and the ever-increasing number of sources of data? And the way we frame the question and the way we've tried to present solutions is around the following. How do we bridge the gap across an ever-expanding and growing number of data sources and leverage that information real time to bring that information to bear on clinical trial decisions? And it is this question that we will focus on, in particular, in the latter two-thirds of this presentation. First, a brief introduction. There, of course, has been increasing industry and regulatory interest in experimenting with more flexible clinical trial designs. And not only are decentralized clinical trials a part of this paradigm of more flexible clinical trial designs, so too are historical control studies, studies that reference real-world data and real-world evidence to provide additional supportive information in a clinical trial, non-concurrent controls, et cetera. Again, there's an increase in flexibility from regulatory bodies and more interest in industry in moving into non-traditional study designs. Of course, the COVID pandemic has accelerated interest in particular, in the decentralized trial design. There is an absence of US FDA regulatory guidance on the topic, though I will note, and I'll talk about this ever so briefly in just a minute or two, there was guidance planned to be produced on this topic for 2020. It is yet to be known whether or not that guidance will still be forthcoming, given their increased focus on guidances around handling clinical trials during the COVID pandemic. There are, of course, numerous considerations around designing and executing a decentralized trial. Derek and I are not going to focus on some of those early aspects and determining the viability of such a design. Again, it's going to be more in the later stages of executing such design and the value add that we think we can and should extract there. We're really going to focus on the space that we operate in, which is partnering with biotech, small pharma, companies, organizations that may not have the depth, the horsepower to take full advantage of these new trial designs, the volume of data that are coming out of these trial designs, and so on. So we've broken the webinar up into three pieces. The first will be an overview. I'm going to spend maybe 5 or so minutes stepping through some of the very basics of decentralized clinical trials, along with some terminology, just so that we're all operating with the same foundational framework. The bulk of the discussion will be in parts 2 and 3. Derek will pick up and describe some case studies that we've, actual studies we've supported, solutions we've presented, challenges we've had to overcome in the decentralized clinical trial setting. And then part 3 will be where we present sort of the foundation for the solutions that we are developing, as well as some actual examples that have implemented some of the ideas we will talk about. So again, the bulk of the discussion is in parts 2 and 3. So a little bit of overview. Let me start with terminology so that the language I'm using is clear and hopefully understood by everyone. So I think everyone has a sense of what a traditional clinical trial is. And from a data collection standpoint, it's data collected at a designated research facility or perhaps through a CRO. But the important point here is there is face-to-face, site-level interaction with patients who have engaged in these clinical studies. A decentralized clinical trial, on the other hand, involves data collection at remote locations. It could be done via, likely is done via technology, et cetera, but the requirement that data be collected face-to-face via, site coordinator interaction, et cetera, is removed. Decentralized trials may use telemedicine, a topic that's been of great interest here over the last several months. Certainly will involve digital or remote technology. This is where Derek and I will spend a fair amount of our time discussing challenges and solutions, electronic informed consent, and so on. The digital remote technology captures endpoint data directly, either via wearables or, we could be collecting patient diary information, eCOA, ePro, and so on. I'm going to, the next couple of the slides, I'm going to cover a little bit of terminology here and make sure this is clear. Hopefully this will be clear to everyone. Fully decentralized trials, which are not the norm, by the way, in this space, are when all activities occur outside of a physical study site. A hybrid study, which is what we have more experience with, and generally where the industry is taking, sort of smaller steps into the space, are when data are collected remote. Some of the data are collected remotely, but some data, some interaction still takes place at a study site, at a hospital, a clinic, what have you. Again, the former example, we aren't seeing quite as much activity yet. There's been a lot of publicity around the handful of those that have been run. More often than not, sponsors are engaging in a hybrid type design. There is some overlapping terminology out there, and let me just take a minute to define a few of these terms. And I'm going to use the FDA's terminology here. So while there is conflicting and overlapping and synonymous use of these terms across the industry, FDA's put down a few guardrails here, and that's what I'll try to stick to here. Virtual trials and decentralized trials in their paradigm and their vernacular are not the same thing. A virtual trial is one that takes place in a preclinical setting and typically involves modeling of patient outcomes rather than the engagement of human beings in an actual clinical study. A synthetic clinical trial actually has gained a lot of popularity over the last handful of years, in particular since the release of the FDA's framework on real-world data, real-world evidence. And this is where non-traditional control arms, for example, can be used to supplement data or can be used as the only source of data to add a perspective to clinical trial results. So this is using electronic health data to act as a control arm, to inform a control arm, or other real-world data, real-world evidence, et cetera. And then finally, historical controls are a setting in which, generally speaking, patients receive an active therapy in a clinical study, and the comparison against which those results are compared is an arm from a previously conducted clinical trial, possibly a placebo arm, another active arm, et cetera. But there's a comparison made using appropriate statistical methodology to say something about the effective treatment. So while these three terms may be used, sometimes overlapping, sometimes not, I think it's good to lay that out clearly. Let me quickly run through the industry and regulatory landscape. So from an industry perspective, as I've mentioned, we've been experimenting with non-traditional trial designs at a higher rate over the last half decade or so. In particular, in the rare disease space, the FDA, from a regulatory perspective, has made great headway in trying to be more flexible in the rare disease orphan type space. This was an evolutionary process pre-COVID, post-COVID, and as we're in this pandemic, certainly this has accelerated rapidly and hence has gotten our attention. We've sped up our development efforts in this area. There are no regulatory guidance documents that directly address decentralized clinical trials, and this, of course, is a point of concern for many of our partners as well as for us. Let me quickly talk about the regulatory documents, and there likely are others that have informed some of our decisions on decentralized clinical trial execution. There certainly is the framework for FDA's real-world evidence program. There are a number of guidance documents and updated documents on conduct of clinical trials during the COVID pandemic, and quite a number of discussions, I think, to give us some early vision into FDA's thinking on decentralized trials with respect to how do we conduct trials, how do we adjust trials in a decentralized setting. Again, they had planned, there was an agenda item from FDA to release a guidance document on the topic. And then finally, GCP, of course, in 21 CFR Part 11, cover all topics related to data collection. So just good to keep in mind. As we're designing these trials, a couple of big topics to consider: the viability of the trial and then the data quality and integrity. Not going to spend much time here. From a trial viability perspective, we have to, of course, consider patient safety, the disease under study, whether or not the investigational product can be provided and administered remotely, so on and so forth. And then from a data quality and integrity perspective, which is where Derek's going to spend his time, we have to consider a number of these topics. The actual technology itself, whether or not we've got a well-founded RBQM system, what kind of data monitoring processes are in place, and data integration and visualization capabilities. Benefits of decentralized clinical trials are, of course, numerous, and they've been well documented. There are many thoughts here around why this is beneficial to patient recruitment, patient retention, a number of papers written on why this may increase patient diversity in the population that's targeted for the particular therapy. Where we're going to spend our time is on the additional data that are collected and the volumes and masses of data that are collected and the value add we think we can have there. Broken up the considerations kind of into 5 buckets here, which I won't read off, but what we found is that there's our sponsor partners have spent a lot of time thinking about the first couple of buckets. Is this a viable option? Is this good for patients? Can we make this work? And where we feel like we've had to fill in the gaps and be out in front, sort of the tip of the spear on many of these topics, is with respect to technology, the data collection methods, the actual data quality and integrity of the overall clinical trial data. In other words, are we producing a study and results that are both internally and externally valid? And then finally, data amalgamation and visualization capabilities. All 3 of those topics we feel like we've had to be out in front on. Some of the questions and issues we need to address in each of those areas: data being fit for purpose, whether or not we're being 21 CFR Part 11 compliant, usual sets of validation and for data traceability issues there. And then, again, topics related to data amalgamation and visualization. And we think this is where the CRO value add is. Derek, I'm going to go ahead and turn it over to you at this point. And I got ahead of myself there, sorry. And let you take this.
I think we're
the role of data management.
Yeah, thanks, Rob. Rob and I are fighting over control of the slides right now. So like Rob said, my name's Derek Lawrence. I'm the Associate Director of Clinical Data Management at RO, and I'm going to be picking up the webinar from this point on and really kind of focusing down on the data management piece among some of the other functional areas, the role it plays, the disruption that the introduction of these more patient-centric, much more effective, methods for capturing high-quality data directly from a subject is really having on the industry as a whole and specifically in clinical research. So just to kind of go over, data management for most folks that aren't in the functional area, a lot of the time is very much a black box. So in kind of a quick oral history of data management, when clinical research was being conducted 20 or 30 years ago, most of all of the studies were done on paper. And so these massive amounts of paper, so all the data that were actually captured at the site were basically sent to a central location and were hand keyed in to some kind of database. So, and these folks were actually represent the first data management personnel, so double data entry staff. Evolving from there, we decided to skip the middleman and give the sites direct access to the databases that we were actually entering the data into in the first place. And so we'd like to talk about the EDC revolution was kind of born. And it was less so a revolution and more just we took what was on paper and we put it in a computer. In retrospect, there were some things we could have done differently, but EDC systems over the past 20 years have really been the predominant source of data capture for most clinical trials. Now, that being the predominant source, 90+% of all data for your standard clinical trial in a non-decentralized fashion is really being filtered in through that single point of contact. So all of it's coming into a carefully constructed EDC system where we're applying multivariate edit checks, validations, we're confirming the presence or absence of data, and around those systems really plug in project management personnel, clinical operations personnel, and they're all workflows built into these systems that support the other functional areas. So cutting to the chase, the landscape is changing wildly fast in that more and more trial data are captured in non-EDC systems. So with the number of technology solutions that are available today, we have everything from patient-reported outcomes and clinical outcome assessments that can be collected directly from the source, biosensors, wearables, environmental data, potentially even social media. We haven't, the viability of a lot of that data has limited utility, but it's still being done in parts of the industry. And all of this, direct-to-patient capture, device collections, and really the increasing prevalence of eSource are not just disrupting data management. It's not just hitting data management that we have to deal in more systems with different configurations, with different kinds of outputs, but it really affects the processes and the workflow that have been constructed for other functional areas, including ClinOps, project management, statistical programming. On top of all of this, the FDA's second revision to E6 really shifted the burden for conducting risk-based quality management to sponsors. Now, while that responsibility can be basically signed over to a CRO, like Roe or other organizations to conduct, CROs and sponsors need to be ready to pick up the responsibilities. So just as a quick primer for E6R2, because it does play a pretty heavy role in the rest of the webinar, it's not just a single aspect of development, execution, and maintenance of trials, but a larger and very multifaceted framework. And some of the high notes for the requirements of E6R2 are centralized documentation of study risks, tracking of study issues, actions, and resolutions, having a clear path of escalation for those identified study issues, and then, shifting more onto the technical side, reduced or targeted SDV, more data-driven approaches for the conduct of on-site monitoring, and then centralized off-site monitoring. And also, as Rob mentioned earlier, more advanced data analytic and data visualization tools that'll help us really pull apart the data and surface data trend or quality concerns that we have. So the biggest piece that's impacting data management right now are kind of the holy trinity of data source diversity, volume, and velocity. And so the one sentence primer there is just that we have data of a much greater size coming in significantly faster from more sources than we ever have before in data management's history. And the disruption is occurring there in the fact that the current model for data management is extremely linear, and it really depends heavily on a tried and true sequence of events and kind of rigid structure to help facilitate the cleaning and the maintenance of that clinical data. And again, anytime we're working in a system that has a single input, it's much easier to predict problems or make forward-looking plans or apply risk mitigation. So some of those pieces are really the existence of clinical sites overall, a separate source in addition to the data that we're collecting in EDC, and that can either be electronic health records, paper backups, et cetera. And through that, it also gives us the ability to query if the data don't look quite like what we're expecting. They produce a digestible amount of data, as we're typically capturing data at individual study visits where clinic staff are, either applying assessments or they're capturing, biological measurements, and those are done at pre-specified time points. Layered around all of this is kind of a listing-based approach to review. We set some kind of a threshold for what we deem to be anomalous, and then we basically create output that shows us values that don't seem to fit based on some arbitrary cutoff that we've established. And so increasing the number of inputs to the clinical data management sphere really adds additional layers of complexity to doing everything from assessing risk, detecting potentially anomalous or erroneous, data values or subjects or problematic sites, and really establishing a clear path of escalation. So, and as Rob mentioned, the pace of change while it's sped up wildly here in the past, few months, due largely to COVID, which has, kind of expedited the need for our adoption of a lot of these different tools and approaches. Clinical research is a very heavily regulated industry, and it tends to react much slower to change than other industries due to momentum alone. So it's kind of like, you have a large freight train is going to take miles and miles to slow down just due to its weight and momentum and its speed of travel. So a lot of these patient-centric tools and platforms and approaches that we're looking at that are a key piece of decentralized trials significantly decrease patient burden and go to mitigate study risks, but they're not without an increase in disruption because they are so different from the norm. And again, going back to E6R2 and with R3 to come, the third revision to E6 is actually underway, and I think that is it's due to be out in 2021 at some point. But the lesson here is that clinical research overall is still wrestling with these challenges that are basically presented in the second revision, which was actually released in 2016. Excuse me, a recent poll that was conducted, I think it was 2.5 or 3 months ago, indicated that only 8% of companies out of 188 polled have actually successfully completed E6R2 adoption, with 17% of the 188 that were surveyed less than halfway there. And that's not a great adoption rate 4 years later. Some of the top problems that were reported, which again all really play a central role in the conduct and maintenance of decentralized clinical trials, are approaches to risk-based management, quality tolerance limit implementation, which is a piece that Rob will get into later in the presentation. Conducting effective change management. So how do we basically fit the new into our existing processes? And also vendor oversight. Again, with that proliferation of data sources, we're dealing with more and more data inputs that have to be managed in different ways than the standard EDC kind of paradigm has allowed in the past. And before we get on to the case studies, I think the biggest piece to hit on that's not a piece of technical input is moving forward the importance of cross-functional collaboration. Now, clinical research and the different functional areas within it has been extraordinarily siloed in the past. And these vertical silos have really hindered our ability to work cross-functionally in the past. Now, when more processes and approaches were very manual, the silos didn't slow down work too savagely. But now that there are functional areas that have previously been relying on these manual processes are being rapidly driven into kind of data-driven processes and the adoption of new tools and technologies, this immediate, early, and frequent cross-functional collaboration becomes far more important. And biometrics groups, specifically data management, statistical programming, and biostatistics, really need to operate in tandem and integrate with clinical operations and project management functional areas very early in the process. So moving on to a few case studies. So that's kind of the high-level overview, but what does this actually mean? So what does the old look like against the new? And I think the two easy examples that we have experience with at RO that do a really good job highlighting the differences in management styles, effective protocols and procedures, and really the challenges that are presented by the new tools are really patient-reported outcomes. So we have ePro, and then we have device data collection. So moving to the first example. So again, years past or fairly recently, diaries were collected from subjects on paper. So basically they were sent home after being enrolled in a particular trial with a diary where they were going to keep track of everything from nutritional intake to pain scores. And basically the sites and the site staff that were present at the time provided an initial layer of kind of education and error detection. And then the subjects are, the patients are instructed to, complete these diaries, and at regular study visits, regular intervals, they would come back into the clinic, and this layer, the interaction with the site staff, provided multiple opportunities at regular intervals for retraining. So if there were issues with compliance, they were being seen, they were surfaced early, and we weren't really dependent on the EDC system to surface those compliance issues because EDC was not the source. EDC was functionally the place where the data were being transcribed. In addition to that separate source, the processes that are kind of folded into this are regular interim monitoring visits by CRAs that are conducted to review this data that's transcribed into EDC versus what data are being provided to the sites to guarantee its veracity. And if we have questions just in a completely one-way blind fashion, EDC could always be queried, to send questions to the sites to say, this data just doesn't look quite like what we're expecting. Can you look into this? So let's move into today where we have ePro diaries. And so this can be either a bring your own device style or hybrid where some devices are provisioned and then other subjects are using their own personal device for data collection. So the biggest change here from a structural perspective is that these data are entered directly by the patient with zero intermediaries. So there's no oversight, there's no check-in. The data are entered in a timely fashion and the data are kind of integrated into a database real time. And the application of these is great because of its patient centricity, but if you really effectively fold this into the design of a study, this does mean potentially longer periods of time between site visits and contact with the patients. So you start to lose that the ability to retrain without being heavily into near real-time data. And because it's entered directly and there is no paper source, the data are the data. Meaning if you're not proactively engaged in monitoring the data, cleaning it, reviewing it, you basically are stuck with what you have. There is no ability to go back, there's no source, we can't query it, and it is what it is. Which means it also disrupts the processes conducted by CRAs in on-site monitoring visits because there functionally is nothing to monitor. Now, one big benefit to this is it does remove questions around the timeliness of entry and the truthfulness of the diary data that we get. There's always the suspicion, with diary data, especially if there's, patient compensation involved, that folks are sitting in the parking lot of the clinic if they've forgotten to fill out their diary for the past 2 weeks, kind of furiously transcribing what they best recall to be their pain scores. But things like, ePro and BYOD studies really take those questions out of account because we have time-stamped attributable data to when those were submitted. So moving on to a second case study around devices, and this is where I think things get particularly interesting for those of us on the technical side of things. Basically, for studies that are doing glucose monitoring. So an old method of this were patients would basically use their own monitors and they would test at home at regular intervals and record this on some kind of a diary for eventual transcription to EDC. And with this, you may be taking 3 measurements per day, morning, noon, and night. And, the diary format is simple. It's very row by column. And again, it follows the exact same kind of review and the source data verification protocols as the ePro setup. Regular IMVs kind of give us some insight into how it's going. Do the sites need to be retrained? Do the sites need to be interacting with the subjects differently? And, EDC can be queried if questions arise. Enter the continuous glucose monitor. These devices are worn by the patient and measurements are captured at 5-minute intervals. So the data volume here is potentially massive. So in the first case, if it's a 12-week study with 3 measurements on a daily basis, you're basically looking at around 250 observations per patient. And that's for the entire course of the study. Now, because it's row by column, those data are also human readable, just basically in standard row by column format in raw form. Move to the continuous glucose monitor. Measurements captured every 5 minutes for the same study and the same duration yield about 24,000 observations per patient and over 280 observations per day. Now, each day represents more data collected than the entire study in the previous example. So, and the data format of this is much more complex. Most of these devices are not row by column. And anything from ODM XML to JSON format. And if you open these files up, there's no way that you could just review this data in its raw form. So there need to be kind of more advanced analytic methods to visualize the data and be able to confirm its quality, because we're well beyond listings here. If there are 24,000 observations per patient, what would a listing of potentially erroneous values even look like? So again, the model doesn't even work all that well. So moving on, that's just kind of the description, the technical lay of the land and how the processes are being disrupted. But just wanted to do a quick move into, how best to support or maximize the benefits of decentralized trials. And I think with the change in circumstances, it makes perfect sense to talk about why we need to look into new methods of managing or reviewing data and confirming data quality. And I apologize here. I probably should have added a phonetic pronunciation to this slide. The Welsh are not known for their English pronunciation or translations. But this slide basically represents the Cynefin Framework. And this is basically, it's a nice model to be able to differentiate between simple, complicated, and complex and chaotic systems, but really be able to understand the different ways in which we have to handle them. And so a general description of all of these, just kind of in a quick nutshell, are for obvious or best practice models. The goal is clear and the way to reach it is straightforward. The best ways to deal with it are standards, procedures, and compliance. So moving to complicated, the goal is still very clear and predictable, and there's a proven way of how to get there. So this represents kind of standard practice from probably 5 years ago. Parts of it may be difficult, but with using best practices and conventional project management, it allows you to plan and execute to really reach the target, whatever that happens to be. Moving into complex, this is basically where we're at with decentralized trials that are operating in kind of a hybrid fashion. The goal is pretty clear, but it's probably going to change along the way based on emergent attributes. So based on the ongoing interaction between parts of the system, the outcome isn't necessarily predictable, and typical cause and effect relations really are only perceived after they occur. So the primary way to deal with systems like this is through constant feedback with a common focus on what is crucial in the, what is crucial in the data. And just for the purposes of clarity, I don't think nothing in research we're approximating chaotic. But just, chaotic is, even if the goal's clear, there is zero way to know how to get there. So cause and effect is out of order, and the only way to deal with chaotic systems is through trial and error. So the most important issues to focus on in research are the complicated and complex ones, and as those are the ones that have to, be managed most aggressively. But in today's climate, everything is treated like a complicated system, and that doesn't always yield good results when combined with conventional project management methods. Because traditional project management methods, don't work very well when applied to complex topics. So an easy example just from my own background, I have 2 small children, and my oldest is 7, and he's a very big fan of Legos. So the act of putting together a very large kind of 2,000-piece Lego set is a complicated system. But the interactions I'm having with my 7-year-old, with my 3-month-old daughter, and my wife as I try to put this together, take care of her, and make sure none of the Legos sneak out to where she's going to step on one again, is complex. So basically this boils down to, with all of the changes, all of the technological applications, and all of the associated impacts that we're having, we really need to develop an understanding of the complete data landscape and how best to apply, in a risk-based environment, controls to manage that process now that it is so different from the norm. At this point, I think I'll hand it back to Rob, who's going to do a brief background on kind of the history of statistical quality control.
Yes, thank you, Derek. So I think Derek did a good job of setting up this next segment. This third part of the presentation will really now move into some of the solutions that we have discussed, or at least the foundation for solutions that we are implementing in decentralized control trials. As Derek laid out the problems in a couple of case studies and then the follow-on slides, one of the preeminent problems, of course, is the volume of data, the number of sources of data. And in the old world of data collection, these data were oftentimes, without much intervention, human readable. Quality could be managed without much technical intervention. And we're presented with new challenges where we can't operate in that paradigm anymore. So how do we solve some of those problems? One of the things that Derek and I have discussed is the application and move forward with is the application of statistical quality control ideas in this space. And we think that this can be particularly beneficial, can be helpful in leading to high quality results. Again, data that can be relied upon both internally and externally. So let me lay a quick 3-slide background on the sort of general theory around statistical quality control, and then Derek's going to apply that and show some slides regarding some of the solutions that he and his team have come up with. So just a couple of slides here on this topic. You know, the great thing about moving into this space is we actually don't have to reinvent the wheel on a number of these topics. Industry, both in the U.S. and outside the U.S., have done a great job of developing quality control methods that lead to sort of well-controlled processes and highly reliable data. We can leverage some of that, of course, keeping in mind we're dealing with you know, a clinical setting. This isn't a manufacturing setting, but we can leverage that and produce high-quality results in this new decentralized paradigm. So industrial manufacturing laid the groundwork for a number of statistical quality control ideas. And you can see the parallels here. Statistical quality control is developed because manufacturing systems develop beyond the realm of single individuals. Again, we have a parallel here. And we needed to make assessments on whether processes were in control given large amounts of data. This certainly sounds appealing. There have been a number of tools developed over the last 100 or so years, and we have chosen to leverage some of them. So I'm going to talk about 3 topics here, just very simple ideas of statistics. This is only one slide, and this is it. The idea of rational sampling, that's what Derek and his team are starting to get into, and then some foundational quality control ideas. So just 3 topics here. So 1, the statistical foundation. Although there are other distributions that apply, let me stick with something very simple. With the volume of data that are coming in, the speed with which the data are coming in, we can leverage statistical methodology to help us understand and monitor whether or not a trial, a particular data collection process, even a particular patient or site are under control, or whether or not we need to intervene, examine, retraining, etc. We can rely, when we get volume of this data of this magnitude, on the normal distribution, the central limit theorem, which is this wonderful thing that I won't explain in detail, except that when we start getting large volumes of data, when we sample and average over and over again, it's normally distributed no matter what. It's a wonderful concept that we can build upon. And this, in fact, has manifested itself in industry, and now it's starting to manifest itself in the decentralized clinical trial setting as control charts, these ideas of quality tolerance limits. And we're using some of this methodology to help us inform those quality tolerance limits. So that's topic 1. Topic 2, the idea of rational sampling. So it's important in any setting to understand the sources of variation, what it is we're sampling, whether it's a human, it's a site, something else, whether we're looking at means or we're looking at proportions, things like that, and how often we need to sample. These are all questions that we ask as we're developing tools, developing solutions, and trying to, again, reap the full value of the data coming out of these high volume applications, decentralized applications. Third and final topic I'm going to touch on is related to some general quality control foundational ideas. And I'm going to spend just a moment on this topic because I think it's particularly informative. So Deming, who is known as the father of quality control, laid out 14 sort of management points on quality control, which I am not going to read to you, but I am going to touch on a couple that I think demonstrate the parallels in what Derek and I are trying to pick up from statistical quality control theory and move it directly into applications in decentralized clinical trials. And the 2 are: 1, we need to cease dependence on mass inspection for quality control. So you can think about that as 100% source document verification, things like that. We know we need to step away from that in this new paradigm. The source itself is the wearable or the device, and we need a better way of knowing whether or not we're dealing with something that is yielding reliable results. The other point I'll bring up here is that we need to break down barriers between departments. And this is something that Derek and I have realized in our discussions as we've advanced on this topic. And I think this talk here and some of the solutions you're about to see are a manifestation of that. I'm going to try to put some of this, sort of put a finer point on this, and then Derek will carry it forward from here. It's the idea of the loss function. And I'm going to talk about Jello for just a minute. So if you aren't familiar with the loss function, I think it's a relatively intuitive idea that, I'm not going to describe it in full, but generally as we get further and further outside of tolerance, the loss in terms of loss of information, loss of reliable information, loss of reliable data, that tends to go up as you get further out of tolerance. And I think that's sort of an intuitive idea. I'm going to tell you the Jello story, and I think you'll see what the parallel here is to clinical trials and what we're doing in the decentralized setting. So the interesting thing about Jello is it has very narrow tolerance limits. So as long as Jello and General Foods is being produced in a very, very narrow window of tolerance limits, we're going to get Jello when we add water to it. What's interesting is when you get outside of these well-defined quality tolerance limits, you actually get either mush or cement. And the loss function is actually very steep with that. It is that general concept that Derek and I are applying to, as well as the entire RO team, are applying to our decentralized clinical trial monitoring, data amalgamation, data summary for us to have a good handle on the current status of data, the tolerance of the data, as well as whether or not we're getting out of that tolerance. So I think my general storyline is we figured this out with Jello. We can certainly figure this out in the more important setting of decentralized clinical trials. So Derek, with the Jello handoff, I'll let you pick up some of our solutions.
All right, thanks, Rob. So picking kind of tying a bow on the first two sections of the presentation again, I think it's been interesting that we've already seen documented struggles within the industry of organizations, sponsors, and CROs having trouble really adopting the risk-based approach because it is so fundamentally different to kind of the linear pre-planned approach that we've been using in clinical research for the last couple of decades. So really talking about where a CRO can operate in the solution space to offer a fix for some of these things that are plaguing sponsors right now. So just a quick overview of kind of the potential benefits of the decentralized trial setting. Basically, it boils down to reactive versus proactive. So the current clinical research model is extraordinarily reactive. So knowing that we have a paper source, knowing that every site will get interim monitoring visits, every piece of data will be looked at on paper and looked at on the EDC, and we will monitor all of it. It's extraordinarily reactive. So we see issues coming out of data that we're exporting from an EDC system, and then we have to go back into it. We're like, we're waiting to see what comes into EDC before we take any action. When we decentralize trials, when we move to more direct-to-patient data capture, when we diversify all of our sources and we can't count on EDC in the same way that we have in the past, we have to have a process, a very thoughtful process for bringing issues to the forefront sooner that might otherwise be missed. I think the best example of this is really talking ePro. If you don't monitor the ePro closely, and there are thresholds for statistical analysis for analyzable weeks, and you don't look at that data until halfway through the study, if subject compliance is terrible and there has been no effort to get that data in the hands of clinical operations or the sites, if there are sites, to fix the issue, you might have just basically sealed the fate of that study's ability to analyze any of the ePro data. We have to apply very rigorous, proactive risk mitigation procedures and controls and have a data-driven way to monitor those. Again, they have to be very rigorous and they have to improve trial quality and data quality. They have to be able to move the needle. So again, per E6R2, the focus has to be on what is critical to the data. We can't kind of globally consider everything of equal importance. Again, it also allows us to identify needed training to implement sooner, and it really improves monitoring efficiency by using data-driven thresholds. So why spend 3 visits a year at 10 sites out of a 30-site trial that are absolute gold star sites that are having no issues? Wouldn't it be more beneficial to spend more time at the sites that are struggling or those that are having issues with specific patients than to just globally spread it around everywhere? And I think that's the underlying concept that decentralized trials really need in order to be successful. So again, just as what Rob was talking about with Schuhart and Deming, basically the plan, do, act, check cycle, RBQM operates in functionally the exact same way. We're going to start, this basic chart breakdown just shows you're going to identify critical processes and data. After that, you're going to identify risks, assess each risk to see how critical it is, each individual one is to the study conduct, and then decide on controls to apply. And then after that comes the continuous feedback cycle that was described in the Cynefin Framework for complex systems. We are going to constantly be reviewing performance. We will communicate, we will review, we will report, and we will constantly be checking to see how our risk controls are behaving. So again, phase one basically being risk identification and planning, and phase two being that cycle, the cycle of continuous improvement that we put in place. So the question here is, like, what does this look like in practice? How do we compile all these data from these sources? How do we get it visualized using statistical methods in front of the clinical folks who are going to be responsible for monitoring the conduct of this study, either in a central or on an on-site fashion? And what we're looking at here is just an analytics tool dashboard with individual funnel plot and other histogram KRIs that show site performance. And again, the risk overview kind of at the top of this just indicates red obviously being KRIs that are, out of the expected quality tolerance limits. Again, statistically set thresholds against the means for the study where we would assume an AE reporting rate doesn't look correct. Entry timeliness, missed visits, things like this are all being funneled into a single visual interface where we can very quickly kind of get the lay of the land. Because for this, kind of demo example, I can already see too much red. So there's already a lot of inherent risk in whatever this would be showing me. And I can see that without having any baked in really intense statistical knowledge. So moving on, these platforms also help us to look at data from a high level view and see if there are any particular trends that we can identify in particular global studies. So again, this is kind of a histogram heat map that just shows based on the shape size, all the rectangle sizes are number of subjects affected. So a larger rectangle is basically a site that has more subjects. And so the larger the site, the more subjects, and the closer to red it becomes, that trial integrity is being threatened because of an overall violation of several key risk indicator thresholds for a single site that contains a lot of your patient population for a single trial. Is it regional? Are you having issues with one particular country? Are you having issues with Europe, the United States? These kinds of tools in reviewing data in an aggregate fashion with it from a statistical basis allow you to really quickly make these determinations and then get into that cycle of continuous improvement where the feedback is constant. You can track issues that are escalated and follow them through to completion. And again, just other visual functionality here is examining the risk of the study over time. So again, being able to go through and say, is this study are our interventions succeeding or are they failing? And that's a question that pre-E6R2, pre-RBQM has been extraordinarily difficult to answer. We just know per the data something is a problem or it's not an issue. So again, these kind of data-driven RBQM applications allow us to answer a lot of questions that really weren't possible before. And so to kind of wrap all of this up, the solutions to manage the complex are largely threefold, or in Rob and I's kind of experience and Ro's overall experience, it's threefold. It's around the tools and personnel and process. And for tools, really leverage a limited set of thoughtfully selected solutions to develop core expertise, because there's always going to be a new kind of plaything, a new toy that comes through that's, it's going to do absolutely everything for you. But having a modular, flexible approach where you can really apply a small handful of tools leaves an organization very agile in the decentralized clinical trial sphere. So SME input is really critical at the point of configuration development, and we're back to the functional area collaboration. Tool selection is not just something for the technical folks anymore. It's really a cross-functional effort to identify, implement, and maintain tools at an organizational level. Moving on to personnel, we really need to encourage cross-functional skill set development in a lot of different functional areas. So again, clinical operations is getting into more data-driven decision-making, more data systems education. And data management needs to be aware, because of that data source diversification that we talked about earlier, about what are the analysis implications? How do we even go about performing data management functionality in this specific environment? And then finally, process. So really working to develop tool-agnostic approaches to execution in a DCT or an RBQM environment are critical to avoid linking your processes to the specific tool that you are using at the time. And again, the big concern there is that if the tool drives your process, the second the tool is no longer being used, that process is going to age out and it's going to leave you in very uncharted territory, which is, not something we want to be in. So again, to just simplify the overall solutions that we apply, they are focused on tools, personnel, and process. And that actually represents the end of our slide deck and our presentation. I think at this point, we'll turn it over for any questions from those on the line or any Q&A.
Thanks, Derek. So there is still time to submit your questions using the Q&A tab to the left of your screen. We've got a lot of great questions already, and we'll try to get to as many as we can. Okay, so the first question asks, do you feel that any specific reskilling is necessary in order to run a successful DCT?
Rob, did you want to take this from the biostats perspective, and then I can kind of quickly answer it from the DM and system side?
Sure. So I think from the biostat, well, let me first. I think there needs to be an overall shift in the thinking around some of the silos that Derek described earlier. This is a much closer collaboration across disciplines with contributions from several disciplines to create an effective web of systems, web of technology that allows us, again, to harness all of these harness the large volume of data that we are now dealing with. So there is some overlap in having some rudimentary training with data scientists, programmers, with data managers, and likewise, biostatisticians understanding the role of this technology, engaging in product selection, engaging in some of the actual presentations, et cetera, to help inform those, make sure that those are targeted with respect to what's needed for analysis, what's core to the trial, to the protocol, et cetera. So I think the skill itself is more about the cross-functional engagement than it is perhaps in an overwhelming new set of skills. I'll step aside and let you answer, Derek.
Thanks, Rob. I definitely agree with that. The one piece that I would add, and this is more specific, again, I'm just speaking from my own experience. Data management has been a very EDC-centric process for a long time, whereas all of the data that we're really dealing with is coming from a single environment in a predictable format. Now, whether that format happens to be CSVs or text files or SAS data sets, it's all extremely row by column. When we get into understanding the interplay with all of the clinical data that represents what would be used to execute a DCT, there is a huge need to understand kind of the ETL, or the extract, transform, and load requirements for all of these different sources, because they're going to be coming in at different speeds and different volumes, and understanding the implications for having a huge bolus of data, it may be in a device situation, and a relatively limited amount of information coming from an EDC or another system, it really should color how the risk mitigations are applied. So rather than, because I know the advice to everybody 10 years ago was everybody should learn to program. I don't necessarily think everybody needs to learn how to program, but I think a high-level analytics-focused kind of conceptual framework for how data fit together and what are appropriate risk controls not only is valuable to the organization, but it's value from an RBQM standpoint for actually, being proactive in assessing those risks that are going to threaten the trial.
Great. So we've got a bit of a follow-up question here. In your opinion, is having a fully implemented RBQM approach or strategy a prerequisite for running DCTs?
Personally, from where I'm standing, I think it is. Maybe not fully implemented, but I would say more than halfway. For decentralized trials, really, you're really going to, because you're removing a lot of that infrastructure, you need to have a plan for operating outside of the norm in a very risk-focused environment, because otherwise you're going to be spending lots of resources doing a lot of traditional activities on novel data sources, novel systems that don't actually function all that well. Again, they just don't play nicely right now. So I think getting at least halfway or having a solid understanding of the implications for executing a DCT in kind of this E6R2 compliant environment is definitely necessary.
Our next question asks, do you see DCT working well across the therapeutic areas?
Yeah, so Derek, why don't I start and then I'll let you pick up. Is that okay?
Absolutely.
Okay, so I'm not going to give a complete answer, but the answer is possibly not all, and I'll speak from a statistical perspective. When ultimately what we need to produce let me focus on an adequate and well-controlled study setting. Ultimately, what we need to produce are data that are reliable, internally and externally valid. And one of the key components of those outcomes is that we are collecting data in an unbiased way, and we're producing an unbiased estimate of the treatment effect. Now, that's a lot of words, but when we boil that down, when it comes to actually collecting the data, we need to make sure that we have an endpoint that is, one, meaningful and related to actual clinical outcomes. Two is, part of, if it's a scale, part of a validated scale, for example. And then three, we'd like to see something that is relatively objective. And so when we get further away from any of those three things that I just laid out, in particular the third, where we're getting away from outcomes, very hard, concrete outcomes, the less viable a decentralized outcome is. We may need site-level interaction. We may need investigator-related assessments, et cetera. And those are harder to do as we get further away from the clinical site setting. So, Derek, I'll stop there and let you add to that.
Sure. Thanks, Rob. I think the only thing I'd have to add to that is really from the data management side of things, we very much need to get into more statistical thinking and more analytics-based thinking about data that is fit for purpose. And the definition for fit for purpose just being all of the suggestions, the recommendations we would make, or the findings that we have from data that are not perfect. Are they the same as perfect data? And if they are, those data are fit for purpose, with error being kind of inherent in everything. Now, that's been a hard idea to stomach, moving from the EDC environment, where it is there is a source, this goes into EDC, they must be totally logical, they must be in lockstep, and they must be perfect. In decentralized trials, where we're getting a lot of this potentially directly from the patient, where we can't query it, QC it, and devices where we're getting a ton of data, what constitutes analyzability is the primary question that needs to drive, is basically all of our approaches. Are they fit to guaranteeing that the data that we're getting are going to be usable for the purpose in mind?
Thank you, Derek. So this will be our final question. We've had a lot of great questions today, and we couldn't get to them all, but we will do our best to get back to everyone who submitted personally after the webinar. Okay, so the last question is, have you applied any of these tools or processes in practice rather than just sort of at a conceptual level?
Oh, definitely. In fact, the continuous glucose monitor example, and the ePro example for that matter, were both real world live fire row examples. And just for additional color that wasn't necessarily part of the presentation, what made the continuous glucose monitor example even more challenging than the description was that it rather than increase patient burden from having to learn a new monitoring device, they were allowed to use their own. But what that ended up being is there were roughly 10 different kinds of continuous glucose monitors used in a study, and almost every one of them had a different measurement schedule. It had different data formats. Their methods of transfer were different. And so there was a huge piece of ETL burden in getting the data from different sites that had different subjects using completely novel continuous glucose monitors into a single format for analysis. And again, that's one of those kind of like hindsight 2020. You have to be able to weigh the balance between going BYOD and saying whatever you give us is fine, which is okay for a smartphone. But in the case of medical devices or some kind of device capture, you really want to enforce that standardization up front, because all of the headaches and the issues around kind of fitting that data into a single format for use and analysis and cleaning and maintenance really becomes much more difficult.
Great. Thanks, Derek. So I would like to thank you and Rob for participating, as well as everyone for attending this Fierce Life Sciences webinar and submitting so many great questions. I'd also like to thank Ro for presenting today's webinar. As a reminder, the webinar has been recorded, so you will be able to access that recording within 24 hours using the same audience link that you received earlier. Thank you all again for joining, and we look forward to seeing you at future events.